Okay. Um, okay. So thanks for inviting us to give the talk today for the communication with China and British. And today, so in this section, we will talk about the diagnosis and treatment of glioma, glioblastoma in Chinese population, based on multiple dimensional molecular map. And so before we get started, I want to introduce briefly about what the GBM looks like. The GBM is a very severe tumors grows in human brains, and from the CT we can see this is the glioblastoma and what the MRI looks like here. And since the glioblastoma is very badly diseases, and the survival rate for the 5 years survival is less than 6%. So many because of they carries many mutations in the glioblastomas. So this is the statistic analysis from American the central nerve tumors. They all central nerves tumors is like 30% 5 years survival. For the glioblastoma is like 5.6% of survival, right? So in 2005, for the GBM therapeutic way, and they try to using the radiation plus the drug chemotherapy, but the survival rate is only improved from 12 months to 15 months. That's just 3 months better. So the TMZ is the currently is the first line clinical drugs which is used in the clinical medicine. So with since the first since 2005 until now, and the GBM survival rate is still not able to get better. So this is how this survival rate looks like. So basically, we can see the survival rate is like just half patient was dead. This is like 1.2 years, just like that. So the so we wanna compare the in 2000 until 2012 what the sex and different population, what this looks like, the survival rate looks like. Basically, from male and female, there's no significant differences. For the ethnicity, there's not that much differences, and for the age, it's just like two. This is a relative survival, basically from different years old. The survival rate is not varies that much. So from the GBM diagnosis, we can see from the hepatology and CT MRI, and this is hard to diagnosis for the MRI. We have some significance, and from the pathology, they are still hard to differentiate what type of the GBM. Basically, the GBM are different from patient to patient, but from CT, MRI, and histology, it's hard to distinguish. But his pathology may be better. That is why from this diagnosis, there is hard to give us the precise medicine for the GBM. So the difficulties relies on how we are able to define the drugs and what the signaling pathway looks like once after we give the drug, like TMZ. In the clinical, once we give the TMZ drug treatment, most of the patients have the drug resistance. And so what the signaling pathway after the TMZ resistance, and what the signaling pathway if the TMZ is sensitive. So they still need a way to distinguish how this looks like after we give the drug. And so except for the signaling pathway, and we also need the cells space and time after the drug. We need the three-dimensional histology, and also the key molecular expression in different type of cells. And this is also the pre-requirement for the precise medicine. So this is something we wanna to overcome for the precise medicine, especially in this context for the GBM therapy. So this is the paper published in the Cell for the leading edge. They said using the DNA and RNA expression and DNA-seq and RNA sequencing, and this gives us the tool to overcome the mystery of DNA and RNA in the cells. So we want to use this DNA sequencing and RNA-seq to see better about the cells go over. So from the GBM is also is one of the tumors, and the tumor have something in common as well. They have so the heterogeneity is one of the key characteristic of the GBM. This can make the GBM different from others. So that's meaning if you give one drug to treat the GBM, it's not gonna be happen because the tumor cells have the heterogeneity from each other. If you just give one drug, kill one population of the tumor, but the other population of the tumor may have drug resistance, they can resurvival again. So that's why the GBM is very resistant once you give the chemotherapy. And this listed how many the molecule was mutated in the GBM, and they related to many things in the cells, like apoptosis, cell cycle, etc. So GBM molecular characteristic was developed since 2000, 2000 until now, and they like 2006, and people divided GBM into 3 types of subtype. They divided GBM because they want to separate what the main differences of different subtype in order to give better therapy or have better prediction of its prognosis. The molecular subtype of the GBM varies from different group until 2016, and they recognize IDH1 is one of the key factor which can divide the GBM into IDH1 wild type and IDH1 mutant. And these two subtypes have a different response to different drugs. This is just the example, the IDH1 wild type GBM, and what this looks like, and they can also accompany with other pathways like EGF or CDK1, etc. But the IDH mutant glioblastoma, they have additional frequent phenomenon like P53 and RAB1, and this type of tumors have a better prognosis. The IDH1 wild type have the worst prediction prognosis. This is the differences of 2010, and people divided the GBM into these four subtype, and these 4 subtype have varies from the drug resistance, the chemotherapies as well. So currently, we want to expect the precise molecular separation of the GBM can lead and give us the suggestion on how we are going to give the patient the precise medicine. So in this project, we try to using multiple dimensional DNA sequencing and RNA-seq to analyze what the GBM subtype in order to give better better prediction of the patient. And the subject we are using CTGA, which is this GBM patient all from China. These are all Chinese, and our technical map is just like collected multiple dimensional of the data set, including RNA-seq, protein genomics, etc. And we will make all this black box sort of like, and in order to get what type of the drug resistance, and in order to give us the precise medicine in the GBM. Okay, and this is how this data set will go over through how we are going to analyze, and we will mapping all this data set into sort of like one just to organize them all together, leading to the analyze the GBM tumor genesis, progression, etc. So we are using this techniques. We analyze the small fan. We recognize that the small fan is one of the key factor in the PTEN related tumor progression, and this is one objective. Another one, we also identified the PTEN was the key regulator which can separate the glioma patient very nicely. You can see the PTEN wild type and the PTEN mutant, they have very different prognosis, especially in Chinese population, the Chinese person with GBM. This is a TCGA dataset. This is CTGA dataset. Chinese population is separated very nicely. And this is how we are able to analyze. This is we separate PTEN wild type and separate PTEN mutant, and we have this kind of number and this kind of number. We analyze the differences of these two patient RNA, all the RNA analysis data, and we try to manipulate what the risk model, which can predict our patient survival rate. This is how we are able to how we get to the model. This model, it's just like once we know this patient's age, like gender, stage, chemo, etc. We can use this model to predict what this patient survival rate looks like. And we're using this model to predict what the driven gene, which can lead to the tumor progressions. And this is the risk model for the PTEN mutant patient. From this model, we can also find the driven genes for the PTEN mutant patient as well. And this is how we are able to analyze the model was correct, and we compared the PC1, PC2, and PC3, and we can see this reflect each other very nicely. That's meaning the model is right. And this model was also analyzed for the tumor looks like, and it's with the immune system, and what this immune system, and with the PTEN status and risk group, and this is all the data looks like. By using this model, one of the key I will stop here, and that is we're using this model. PTEN mutant is the worst subtype in the GBM. By using this model, we identified that these three gene was the driving gene drive the PTEN mutant tumors to grow very badly. And we're using the drug screening method and to find one of the compound called JQ1 can targeting these three driving gene at the same times. That's why we got this JQ1 and tested in the experiment and found this is the PTEN mutant. This is PTEN mutant cells, and we're using the JQ1 by targeting this PTEN mutant cells. And you can see this four. These cells basically were killed by the JQ1. But in the PTEN, this is the PTEN wild type GBM cells. The JQ1 is not works that much. So I will stop here and willing to take questions. But but it looks like I cannot not hear anything from the meeting side.
Maybe we are running out of time, so maybe only one question. If no question, we will move to next talker.